Today we'll take up the topic cells and organs of the immune system. In that, I'll take up the first topic, that is cells of the immune system. Now, to know the cells of the immune system, you will need a blood smear of a person where you will get circulating RBCs as well as white blood corpuscles. And this slide you are going to prepare in your practical, and the slide preparation is called as slide for differential count of WBC. You can actually classify the WBC based on their structure and morphology and count the number of WBCs that are there in the blood smear and make a total tally of that and find out which population of the WBC is more and which is less. So in this slide, you will see the large number of RBCs are there. They are brownish and they are small, spherical almost, and without nucleus. They are spread out. They are not going to be counted in the immune cells. The major cells that are going to be counted are the WBCs. Now on the top, you'll see a cell which is pigmented red with larger number of granules in its cytoplasm, which are also pigmented red, and that is eosine stain, and that is called as eosinophil. And then immediately coming below that, there are two cells, that is with the nucleus having dumbbell shaped structure. Here in this case, there are about 4 dumbbells, and that dumbbell shaped structure can go on up to 6 dumbbells. And these two cells which are there opposite to each other approximately, they are neutrophils. Okay, and one which is there at about 1 o'clock position on the top of the slide, 1 o'clock position, which is an immature neutrophil. Its nucleus is not yet formed into dumbbells. It is an immature neutrophil. Then coming to the middle part of the slide, there is a irregularly shaped cell which is blue stained with a large nucleus in it, and nucleus is almost like a kidney shape, and that is supposed to be a monocyte. Then on either side of the monocyte, you are seeing two cells where the nucleus is occupying the entire volume of the cell. Nucleus is occupying entire volume of the cell, and hardly you can see the cytoplasm, and those two cells are lymphocytes. But here we will not be able to classify whether it is a B lymphocyte or T lymphocyte. Therefore, in the WBC counting, they are just classified as lymphocytes. If you want to classify them as B lymphocytes and T lymphocytes, then you have to use monoclonal antibody staining technique. And using the receptors of those two cells, like T cell receptor that is supposed to be CD4, and the B cell receptor that is supposed to be Ig alpha or Ig beta, or it could be even IgM. Using the monoclonal antibodies against these receptors, you can classify these lymphocytes into T and B cells. But in the clinical investigation, when a doctor advises you to go for a blood test, the RBC count is done and the WBC count is done. By using this technique only, other than that, they are not going to do the monoclonal antibody technique until unless it is required. So lymphocytes are these. Then at the bottommost part of this slide, you can see blue stained with blue granules. Large number of blue granules are there in that cell that is supposed to be a basophil. Population of basophils in generally in the in the differential count of the slide will be 1 to less than 1%. Similarly, eosinophil count will be anywhere between 1 to 5%. Generally, it is 1 to 2 or 1 to 4. But sometimes it may go up to 5%, or if it exceeds, then it is called as eosinophilia. Most important cells are the neutrophils. Their population goes up whenever you have viral infection or bacterial infection in the body. And the monocytes which are there, and those monocytes are in fact temporarily there in the blood, that is for about 8 hours, then they migrate to the tissue spaces to become the macrophages. The neutrophils will invade the tissue spaces or areas wherever there is inflammation. I've spoken about that in the part of inflammation as extravasation or diapedesis. Now let us classify the immune cells based on their staining property and property of the cytoplasm. They can be classified as agranulocytes, means without granules in the cytoplasm, and granulocytes with granules in the cytoplasm. And those granules will stain differentially. Therefore, it is called as differential count of the WBC. Two stains are used here, acidic and basic stain. And both these stains are responsible for staining the granules as well as the cytoplasm and the nucleus. And the stain is called as Leishman stain. And that stain differentiates the color of the granules as well as the cytoplasm. The granulocytes are neutrophils, eosinophil, basophil, and mast cell. I have not included NK cell here because it doesn't come in the WBC count and it is not abundant in a normal person. So therefore, we will not be considering that now here, but it doesn't mean that it doesn't exist. It is there. It is there, but in the background, and we will talk about that when we are going to talk about the effector response of the immune cells. But for the time being, you keep in mind the granulocytes that are generally seen in the blood smear are neutrophils, eosinophil, basophil, and mast cells are also not seen in the blood picture. They are found attached to the blood vessels. Remember that in the endothelial lining. Whereas the agranulocytes, that is monocytes, which convert themselves into macrophages, and the lymphocytes, which I already told you, they are T and B lymphocytes, but they are classified as lymphocytes. But in the blood picture, you don't get once again dendritic cells. Dendritic cells are also within the tissue spaces. They are another category of the antigen presenting cells, dendritic cells. We'll start first with monocytes. That is generally seen in the blood. And monocytes which are there in the blood, they are temporarily there for about 8 hours. After that, they migrate to the tissue space. Only they will get excited and active only if they come across any pathogen in the blood. And they have less number of pseudopodia. Their size is smaller, kidney-shaped nucleus with the lysosomes which are less in number in the cytoplasm. And the size of the monocyte compared to the macrophage is about 10 times smaller, and the macrophage is 10 times larger than the monocyte. Now see the pseudopodia on the surface of the macrophage, which is larger in size compared to the monocyte, which is smaller. Therefore, phagocytic activity by the monocyte is much lesser compared to the macrophages. When these monocytes, after their time duration is over in the blood, when they migrate to the tissue spaces, they become macrophages. And macrophages have a lot of other functions to do. One, they play a major role in scavenging the tissue spaces as well as the tissue for the dead cells. When they scavenge the tissue spaces for the dead cells, if the tissue cells have died, those dead cells are going to be eaten away by the macrophage and those cells are going to be lysed. Therefore, they are called as scavengers. Macrophages process the dead cells and debris of the dead cells are removed from the body by the macrophages. Then the second important function is those cells encounter the antigens that are there in the tissue spaces which have entered by chance. And these antigens are exogenous origin, though they don't belong to your body, probably of the bacterial origin or probably of viral origin or any other pathogen. And those antigens will be encountered by the macrophage in the tissue spaces. And macrophage generally shows not only the larger size, it has larger number of cellular organelles and tremendous amount of phagocytic activity plus large number of lytic enzymes in the cytoplasm. And as soon as they encounter a foreign antigen other than your own body cells, I had said that they are scavengers. when they are scavenging your own body cells which are dead, they don't get excited, they don't get activated, and they don't show excessive phagocytic activity at that time. They show general normal phagocytic activity. But all these activities, phagocytic lytic enzymes and cytokine production activity increases in the macrophage only when it comes across a foreign antigen or a pathogen. And when it comes across that foreign antigen, it is excited and activated. I'll come to that activation process little later. Before that, we'll see when the macrophages enter the tissue spaces and become the permanent residents in the tissue spaces. They remain there for quite some time, long duration. Nobody knows when the macrophage dies, only when probably it may encounter a toxic bacteria, it may undergo autolysis. I have told you about that process that is called as highly reactive oxygen species are going to be produced, and there is respiratory burst to encounter the pathogen, possibly that will kill the macrophage. But other than that, macrophage have a long lifespan. And in the tissue space, the macrophage, when it is there as a permanent resident in the tissue space, they are named differently. For example, the macrophage that is there in the liver cells is called as Kupffer cells. Kupfer, Kupffer cells. And those which are there in the alveolar region of the lung, they are called as alveolar macrophages. Those which are there in your connective tissue regions, connective tissue, cartilage, connective tissue, okay, and those are called as histiocytes. And those which are there in the bone, not in the bone marrow, in the bone, they are there in the bone, hard structure of the bone, between the vessel, the cavities of the bone, and they are called as osteoclasts. Then in the kidney, they are called as mesangial cells, and in the brain, they are called as microglial cells. Okay, these are the classification of the macrophages. When they are distributed in different tissues, their role is same, but they are named differently. They are meant for scavenging, removing the dead cells, removing the debris of the cells, keep the place clean. Second, they are very important cells of the immune system because they play a role of antigen presentation. Because as soon as they come in contact with any foreign antigen, they phagocytize that antigen, or endocytosis will take place and process the antigen. Okay, so the macrophages have certain receptors on their surfaces. One of the important receptor is C3b receptor. The C3b receptor belongs to the complement and had come across this complement in alternate pathway of complement when I discussed with you in innate immune response. The C3b, when it is attached to the pathogen, the receptor which is present on the macrophage can bind to this C3b and engulf that pathogen which is being bound by the complement, and that process is endocytosis. The second possibility is immunoglobulin G, which is produced as an antibody against the pathogen, can react with the pathogen and form a complex with it as an antigen antibody complex. And when it forms a complex with the antigen, that antibody together with the antigen can be engulfed by the macrophage by endocytosis, and the receptor for the IgG molecule is Fc gamma receptor, because IgG is called as immunoglobulin gamma, IgG, therefore the name IgG. So Fc gamma receptor plays a role in engulfing the antigen antibody complex and process it and destroy it. So there are two receptors which are very key receptors on the macrophage, help in processing the antigen through the endocytic pathway, endocytosis, both these receptors. Then macrophage also shows pinocytosis and phagocytosis. Phagocytosis, that is without receptor mediated intake, where bacteria is going to be taken up directly as shown in this diagram. In the macrophage, in this diagram, you can see the bacteria is being engulfed by the macrophage using its pseudopodia, which will encircle that, and now bacteria is enclosed in a phagosome. As soon as the phagosome is formed, the phagosome fuses with the lysosome to become the phagolysosome. And lysosome has lot of lytic enzymes. And when the macrophage encounters a bacteria, the production of lytic enzymes will increase. And I had told you this mechanism as oxygen independent mechanism. It is an innate ability of the macrophage, and it will help in destruction of the bacteria within the phagosome. And after fragmenting the bacteria into minute to micromolecules, some molecules are going to be charged or loaded on class 2 MHC, and they are going to be exposed outside the macrophage on the class 2 MHC as antigen presentation. So, exogenous antigen, that is bacteria or virus, bacterial toxins, bacterial polysaccharides, bacterial proteins, all that is processed through phagocytosis or endocytosis, whatever the mechanism. Those are exogenous antigens. They are going to be presented on class 2 MHC of the macrophage. Therefore, it becomes an important antigen presenting cell. When the antigen is presented on the class 2 MHC on its surface, it is going to be recognized by the T helper cell. And without the presentation of antigen on class 2, T helper cell cannot recognize the antigen on its own. So this is one process, and details of this antigen processing and presentation, we'll be talking about that in unit 3. I will be dealing that in detail. Then let us presume that the macrophage encountered a bacteria like the tuberculosis bacteria, or it is another bacteria called as Listeria monocytogenes. There are some bacteria, when they are engulfed by the macrophage, okay, they have an inherent ability of preventing that phagosome from fusing with the lysosome. They will prevent themselves from getting destroyed, and they remain in the phagosome. But the phagosome membrane is permeable for all the nutrients to enter the phagosome. And they take charge of the macrophage as endopathogen. After entering the macrophage, they are not going to be destroyed, but they will remain in the macrophage as endopathogen. And when they remain there as endopathogen, they can multiply there within the phagosome and make the macrophage increase in size, and that macrophage now becomes totally unable to process other antigens. It will paralyze that macrophage, and that macrophage will settle down in a place, and such macrophages which are carrying the endopathogen will become large mass of aggregation in one area, and that is called as granuloma. Granuloma, granuloma. And this is what you see in the TB patients. In the TB patients, the alveolar macrophages which are there in the lung, if the tuberculosis bacteria has colonized the lungs of the person, these alveolar macrophages will engulf the tuberculosis bacteria, and they form a large congregation of macrophages in the lung, and they appear as granuloma. That is what you see as a white patch in the lung. But don't misunderstand, TB is not restricted only to the lung. One can get bone TB, one can get intestinal TB, one can get liver TB, like that, even brain. All different organs can have the infection of the tuberculosis bacteria. And wherever the tuberculosis bacteria resides, and it is processed by the macrophage and remains there in the macrophage, in its cytoplasm, makes that macrophage useless for other immune purposes, and it will prevent the lysosome from fusing, and it becomes a reservoir. Macrophage becomes a reservoir for the multiplication of tuberculosis bacteria, and it will spread from there to other regions if the macrophage undergoes lysis. Unknown reasons. Therefore, when the drugs are targeted for tuberculosis bacteria, those drugs have to be targeted to reach the cytoplasm of the macrophage and kill the tuberculosis bacteria. That's very, very exceptional case. Okay. But generally, normal process is that macrophage is capable of processing those antigens, that is the exogenous antigen of bacterial origin or viral origin, and present it on the class 2 MHC. But when the bacteria is residing within the macrophage, it will behave as though it is the part and parcel of the macrophage. And then, by chance, if some of the antigens of that bacteria are processed, because like the normal cell processes the other antigen, that endogenous antigen will be presented on class 1 MHC. Please remember two things. Those antigens that are produced within the cell are going to be presented on class 1 MHC, and those which are processed from outside, they are presented on class 2 MHC. So the macrophage has the ability of expression of both class 1 and class 2 MHC. This is the characteristic, very, very special characteristic of an antigen presenting cell. Presentation of antigens on both class 1 and class 2 MHC is the characteristic of antigen presenting cell. No other cell in the body has this ability. Okay, then what are the cytokines that are going to be produced by the macrophage when it comes in contact with a foreign antigen? Foreign antigen, I can call that as also as non-self. Self that belongs to you, non-self which does not belong to you, which has entered your body. And when a macrophage encounters a non-self antigen, it is activated. When it is activated, the first cytokine it is going to synthesize is interleukin 1, IL-1. And that interleukin 1 is called as a warning cytokine. As soon as it is secreted, the Th cells that are there in the thymus, the organ, which we'll be talking about that in lymphoid organ, in the organs of the immune system, the Th cell is going to be warned and it is going to be activated, stating that there is some foreign invasion in the body. Now you start your activity. That is the warning cytokine for Th cell. And without the IL-1, Th cells are not going to be activated. If the IL-1 is synthesized, that means the macrophage has come in contact with the foreign antigen. Then the next thing is that macrophage is going to present that antigen on the class 2 MHC to the Th cell, and that we'll see later. But for the time being, you keep in mind, interleukin 1 is a warning cytokine which is secreted by the macrophage when it encounters a foreign antigen of bacterial or viral origin. Then comes the another cytokine, that is interferon alpha. I've spoken about this in innate immune response. It is the innate ability of the macrophage to secrete this cytokine in large quantities and pour it out to warn other cells to resist the viral infection. So interferon alpha gets the name because it interferes with the viral infection, and it is produced in large quantities even when the bacteria is encountered by the macrophage. And this interferon alpha gives boost to the immune system to guard themselves against the infection. Then comes the another factor called as tumor necrosis factor alpha, TNF alpha. Its main aim is to kill the tumor cells, but only when the tumors occur, it will kill the tumor cells. But when it is produced as a cytokine, when there is bacterial infection or viral infection, I told you that it is going to cause high inflammation. It is an inflammatory cytokine. The name it got, why? Because it was first discovered in macrophages that have invaded a cancer region of the body and killing the cancer cells. Please remember, whenever the cancer occurs in the body of an individual, it is being continuously attacked by the cells that kill them, that is macrophage, natural killer cells, and CD8 T cells, which are cytotoxic T cells. These are the cells that are responsible for killing tumor cells, yet the cancer succeeds. Why it succeeds, we will talk about that in Unit 5. But for the time being, keep in mind the tumor necrosis factor that is secreted by the macrophage is an inflammatory cytokine responsible for septicemia and toxemia. In abundance, it is very, very toxic. But in limited quantity, it is good. It is responsible for bringing about killing of the cells. Okay, it is responsible for killing even bacterial cell because tumor necrosis for alpha invites natural killer cells to that area, and it will invite even the macrophages to that area to kill the bacteria. Well, next, other cytokines are interleukin 6. I had mentioned about this interleukin 6. It is responsible for causing fever. When the infection has taken place in the body, macrophage is activated, it has engulfed the bacteria, it has engulfed the virus. As soon as it has engulfed the virus, the activation has taken place and IL-6 is secreted. IL-1, anyway, it is secreted, TH cells are activated. IL-6 is very, very important because it is going to activate the B lymphocytes. The B lymphocytes which are there for the antibody production, they are also now being warned that they have to get ready to start producing the antibody against the antigen that has invaded the body. So interleukin 6 is also called as B cell differentiation factor, BCDF, B cell differentiation factor. It helps for the B lymphocytes to produce the antibodies responsible for tackling the antigen. But at the same time, it goes and binds to the hypothalamus in the brain. It can cross blood-brain barrier and go to the hypothalamus and bind there and alter our regulation of body temperature. And therefore, we get fever. It's a good sign. When you get the fever in the beginning, it's a good sign that your immune system is working, it is responding, and you are developing the immunity against a particular pathogen. And it is a sign that you have been infected. Okay. What is GMCSF? G for granulo, M for mono. So granulocyte, monocyte, colony stimulating factor, GMCSF. Granulocyte, monocyte, colony stimulating factor. This growth factor, when it is secreted by the macrophage, it goes to the bone marrow and makes the bone marrow to produce large number of monocytes as well as large number of granulocytes, which are required now for the defense. Because there is an infection, you need the population of these cells in large numbers. That is why it is secreted by the macrophage. Then another cytokine is called as granulocyte colony stimulating factor, responsible for all the types of granulocytes, including the NK cells production. Then comes a monocyte alone colony stimulating factor. The monocyte colony stimulating factor increase the population of monocytes. So these growth factors are secreted by the macrophage when it is activated. Okay, so the macrophage, which is the tissue residing monocyte, is nothing but a special type of immune cell responsible for scavenging the dead cells as well as processing the pathogens that enter the tissue spaces. And it is responsible for killing of the bacteria using the reactive oxygen species. I've dealt with that example, and it is also capable of killing the bacteria using oxygen independent mechanism. So macrophages are the primary immune cells responsible for the antigen processing. They are also responsible for antigen presentation. And when they present the antigen, that antigen is going to be recognized by the Th cells. That is about macrophages. Then comes the another type of non-granular or agranular immune cells, and they are dendritic cells. They are not seen freely in the blood. They are present in almost all tissues spread out. And since they resemble the nerve cell in structure with dendrites, nerve cells are supposed to be having dendrites, that is projections on their surfaces as nerve fibers. Nerve fibers, those are called as dendrites. Therefore, the resemblance is to the nerve cells and to the dendrites. Therefore, they got the name as dendritic cells. But the dendritic cells have similar properties like that of the macrophages. Their shape and size is different. They are with large number of projections, and these projections which are there are nothing but they are filamentous pseudopodia. They are just like flagella or filamentous pseudopodia. They are not as broad as pseudopodia in the case of macrophages. And these filamentous pseudopodia are extended to a large distance inside the tissue spaces. Therefore, a dendritic cell can trap an antigen on this filamentous pseudopodia at quite a long distance and then receive that antigen and process that antigen, and they also become antigen processing cells. Dendritic cells are also called as antigen processing cells. They have their common origin together with the monocytes, therefore they share the same receptors that are there in the monocytes or the macrophages. They also share the same receptors. And when they process the antigen, same manner as macrophage did exogenous antigen, when it is processed, they will present it on the class 2 MHC. And the endogenous antigen, when they encounter and within their cytoplasm, the antigen is present, and that antigen will be presented on class 1 MHC. So these are also called as professional antigen presenting cells. Their duty is only production of the class 2 and class 1 MHC and present the antigens. Unlike the macrophages, dendritic cells are not known to produce large number of cytokines. They are not known to produce large number of cytokines. They are only meant for processing the antigen. So the dendritic cells, like macrophages, can be residing in different tissues. And depending on which tissues that they are residing, they have been classified. And those dendritic cells which are present in the skin layer, that is in your epidermis of the skin. Skin has two layers. One is epidermis, that is a thick upper layer. Then following that at the bottom, there is dermis. And dermis is the one which is almost very close to your body muscles. So in the epidermal layer, which is multi-layered structure, all epidermal cells which are there, which are epithelial cells, and in the epithelial cells of the epidermis, these dendritic cells are distributed. And they are called as Langerhans cells. You might have heard about this name. It is named after the person, the Langerhans cells. And this Langerhans cells are different from the Langerhans cells that you see in pancreas. Langerhans was the one who had found cells in pancreas also. Alpha and beta cells of the pancreas are also called as Langerhans cells, which are responsible for secreting the hormones like insulin and glucagon. They are also called as Langerhans cells. They are not dendritic cells. Please do not confuse this with that. But the scientist is the same. So Langerhans cells found in the skin are dendritic cells. Then comes interdigitating dendritic cells. What is meant by interdigitating? Digit is nothing but your fingers. These dendritic cells will have finger-like projections on their surface. And those finger-like projections will resemble the finger-like projections of the macrophage. As seen in the slide now, on the macrophage, those finger-like projections are there, which are nothing but they are pseudopodia. Filamentous pseudopodia are different. That is seen in this picture of the dendritic cell where they are fibrous and filamentous. In addition to that, if the dendritic cell possesses finger-like projections on their surface, and they are called as interdigitating dendritic cells. And they are found in the lymph nodes as well as in the thymus, in the thymic medulla, in the thymus, and the lymph node, they are present, and they are responsible for presentation of the antigens to the T lymphocytes as well as to the B lymphocytes. They are interdigitating dendritic cells. Next comes interstitial dendritic cells. Interstitial dendritic cells are found in almost all other organs except the skin. They are there in the heart, they are there in the lung, liver, kidney, and gastrointestinal tract, and everywhere they are spread out in the tissue. They are interstitial dendritic cells. Not much different from either the Langerhans cells or the interdigitating cells. It is just that they are present in those tissues, therefore they are called as interstitial dendritic cells. Then, sometimes these dendritic cells take a decision to migrate from one tissue area. Let us presume that Langerhans cell found in the skin, okay, it encounters an antigen because the skin is punctured now. It has got a wound, and through the wound the bacterial infection has taken place, and that bacteria is being recognized by the dendritic cell, and that dendritic cell now processes that bacteria, and it can remain in the skin and send signals. But generally what they do, because dendritic cells are not capable of producing large number of cytokines, as soon as they encounter the antigen, they engulf the antigen and they start migrating from the tissue to the nearest lymph node. Wherever the lymph node is present in the body, nearest lymph node, they will migrate. When they are going to migrate, they should leave the tissue and enter the blood stream. And when they enter the bloodstream, those dendritic cells which have entered the bloodstream carrying the antigen within them or on their surface are called as wheeled cells. Please see that is shown in blue font, wheeled cells, spelling VEILED, wheeled cells. They reside in the blood for a temporary duration. They are passing through now. They have taken the blood route to reach the nearest lymph node. And once they encounter the lymph node, because lymph nodes are going to be drained by the blood, blood enters the lymph node and passes out of the lymph node in different regions of the body. As these dendritic cells from the blood, wheeled cells from the blood, enter the lymph node, they take the residence there. Now they are responsible for processing that antigen and present it to TH cells and the B cells. In the lymph node, now the activity starts. So the wheeled cells are nothing but traveling dendritic cells from any tissue which has encountered an antigen, and they leave the tissue. Why? Because they have to now go to the nearest lymph node to stimulate the immune response. Okay, I had mentioned that the dendritic cells also express class 2 MHC, and class 2 MHC is majorly responsible for presentation of the exogenous antigen, and this class 2 MHC helps in presenting the antigens to the T helper cells. Then comes another special, very special category of dendritic cells, which are called as follicular dendritic cells. And these are highly restricted. They are not seen in other tissues. They are not seen in liver, lung, and all these areas, neither in the skin. They are seen only in the lymphoid follicular areas in the lymphoid organs. The lymphoid organs are primary and secondary lymphoid organs. We'll talk about those little later. One of the lymphoid organ is your thymus. Second lymphoid organ is bone marrow. They are two considered to be primary lymphoid organs. Then comes the lymph nodes in your body. Different regions of the body have lymph nodes. I will show you the picture of that in the later lectures. And that is secondary lymphoid organ. Lymph nodes are secondary lymphoid organ. Spleen is another lymphoid organ. You have sometimes heard about tonsils, which is a lymphoid organ which is situated near your throat, tonsils. Then you have adenoids, which are on either side of your nasal region, just below the eyes, in that region which has a little bit of cheek cavity. There, the adenoids are there. Sometimes they are painful when you have extreme cold or influenza virus infection. They have they are swollen there, adenoids. So these are all lymphoid organs. And these lymphoid organs will have follicular dendritic cells. What is the speciality of follicular dendritic cells? Unlike the other dendritic cells which I listed, like Langerhans, interdigitating, interstitial, and wheeled cells, these follicular dendritic cells do not express class 2 MHC. They are different. What are they meant for? They are meant for expressing the receptors of Fc gamma, that is for the IgG, and C3b receptor. Therefore, they are capable of endocytosis of the antigen through these receptors and process the antigen. At the same time, when these receptors have been bound by the antigen antibody complex, they are responsible for presentation of that antigen to the B lymphocyte in the lymph node. They're responsible for the activation of the B lymphocyte, for further production of the antibody, and they're also responsible for the production of memory B cells, which will give you long time immune memory. So therefore, these are very important cells found in the lymphoid organs, in the follicles. Therefore, they are called as follicular dendritic cells. So that is as far as today's lecture is concerned.